welcome everybody, and thank you for joining us today. It's a pleasure to see so many of you. First of all, signed up, but are joining us today to talk about soybean gall midge, and I think this highlights the importance this insect plays across the Midwest for all of us. So we appreciate you being here, and I want to reinforce something. I got a couple things to say before we get started, but we titled this a discussion specifically because we want to hear from you. Whether you have questions or comments even, there's a Q&A at the bottom or somewhere on your screen that you can click on. You'll also see one that says chat. We kind of reserve that for technical issues that you might be experiencing and can place things in there. But I have a number of people, we want to thank for helping put this together, and the fact that we have really anything to present to you. And first off is Phyllis Bongard, who's hiding behind the scenes of this whole thing and making us all look good. She has done all the work in collecting your registration and putting up the website, that nice music rollover that we had to begin with, so you can see some of the upcoming events, and as well as the CCA credits, along with Bruce, who'll be talking later today. So thank you to her, especially for all the work that she's done, and she'll be providing us with some closed captioning to follow up with this, so this can be posted for future viewing. Of course, none of what we would be presenting would be possible without the generous funding that we have received from a number of groups, and you saw a slide, and unfortunately I don't have the time to list everybody. All are crucial to what we do with soybean gall midge. A special thanks to the North Central Soybean Research Program, which was one of the first groups to rapidly respond to this back in 2018, provide us some quick funding and continued support. So those soybean farmers that are out there, thank you, Nebraska soybean, Iowa soybean, Minnesota soybean. Thank them for continuing to support this and push this up at a regional level. A special thanks to the soybean farmers. I see a number on here that I work with. I appreciate you joining, as well as the others. I'm sure see some familiar names. Thank you for putting your farms at risk to help us learn more about this. We know it's been challenging. The answers haven't been easy. And so we appreciate you working with us each year to learn a little bit more about this insect. A couple other things is CCA credits or CEUs. There'll be a link provided in the chat that'll have Phyllis has just put up there now. That's a Google sheet to fill out that will provide some information. Then at the end, she will post a QR code, which will help you get those credits faster if you need them. So pay attention to that. I will monitor the chat and the Q&A, especially during the session, provide that back to the speakers, and so we can answer your questions. And I think I've kind of captured everything that we need to cover up front. Any of the speakers, did I miss anything that we needed to cover initially? Okay, so we're going to kick it off and I think this is apparent from what we had on the website, but we're going to kick this off in 12-minute talks, roughly. They're going to vary a little bit. And then we're going to take a break for a few minutes for you to ask questions, post them in the chat, post them in as the speakers are talking if you'd like. And then I will read those to the speakers so they can answer them. Maybe you have something that comes up after day. It's okay. We're going to see one another again in a week here. So you can keep those questions for next week. I know you're going to have the urge, and a lot of you will have the urge to ask ecology and management related questions because that's the applied part of soybean gall midge. In terms of what you might be able to do, hold those, because we want to have a specific discussion on those first on the 12th and 19th. So write those down on a piece of paper, set it aside, and bring those up in the next section. This is probably the most important part, is to do the identification, to be aware of the misidentifications that can occur, and the scouting. And that's really our three kind of key topics today. I think that I don't see any comments or anything from the rest of the panelists. I'll repeat those questions. So we'll start off with Dr. Thomas Hunt. He's going to cover the initial discovery of this insect before we knew what it was, through a bit of what we've seen, and then we're going to transition to Dr. Aaron Hodgson and Bruce Potter at the last there to discuss some of the misidentifications, along with Bob Cook is also here. Dr. Bob Cook The two of them led that effort to identify a closely related species. So Tom, if you want to go ahead and share your screen.
Okay, can everyone see me? Okay, well, hi everyone. I am Tom Hunt. I'm the entomologist at the University of Nebraska Haskell Ag Lab in Northeast Nebraska. And what I'd like to do initially here is give a little bit of background and some basic pest ID as an introduction to our seminar series here, and on our new pest of soybean, the soybean gall midge. So there we go. Oops. Let's jump right into it. This insect was first documented in our CropWatch newsletter in Nebraska, in Northeast Nebraska in 2011. It was observed in late August, and it appeared to be associated at this time with hail injury. We took some of the larvae back to the University of Nebraska entomology department and had them tentatively identified as gall midge larvae. You might ask, well, why didn't you identify them as species? Well, that's because these types of insects require specialists to identify much further than just genus or so. And so we got it to gall midge, and we tried to raise some adults. We couldn't. And so we moved on from there. We assumed them to be feeding on necrotic and decaying tissue or fungi, which is common to gall midges. And so that's kind of where we were in 2011. The first pictures, this is actually pictures from 2010. As you can see here, at the base of the plant, that's very typical of a lesion we would see. This was a hail field. And so we saw these basic lesions at the base of the plant, very typical. Sometimes you'll see an actual gall, but most of the time it's a lesion or a canker. But we also saw some lesions up higher on the plant. And remember, that's because this was a hail field. And I think Justin will probably mention later in the series about how hail plays a role in soybean gall midge infestations. But that's what we saw. And when we peeled these lesions apart, we saw these bright orange midges. Here's another shot, just another shot of this. You see this typical basal lesion. And again, when we peeled it apart, we saw these little orange larvae. Now, you might say right off, well, that lesion, that could be a disease or whatever. And you're right. There are symptoms of diseases that look very much like this. And in fact, we did see gall midges often in association with disease. So that does play a role somehow with gall midges. We don't know quite how. And for the next 5 or 6 years, it was basically that. It was a series of, just a few reports isolated to a few fields. Typically, they were observed later in the season, in August, even late August, and it was assumed to be associated with plant pathogens or mechanical damage like hail, like I saw first in 2010 and 11. And as a matter of fact, a lot of times it was plant pathologists that would bring this up because they'd be out in a field checking a disease problem along an edge of a field, and they'd find these larvae, and they too would assume that these were, like many gall midges, simply feeding on the fungi or the necrotic tissue that was developing because of the disease. However, in 2018, everything blew up. It was getting a little bit more prevalent, but 2018, we had widespread reports of significant injury. Later in the season, you'd see, like in this picture, you'd see some dying plants along edges of the field. And many of these observations were early in the season, these initial observations, like in late June. And remember, before we were seeing them in late August. So this is very different than what we saw previously. And this means there were probably some insects in that field in early June or late May. So this really stimulated interest among the states, Iowa, Nebraska, South Dakota, Minnesota. We started a lot of different projects and research and started talking to growers and soybean boards and things like that to try to get a handle on this situation. So the first step, identification. Like I said, it's tough identifying these guys. You need larvae, and you often need the associated adults in order to actually identify them. And these are the three scenarios that we would typically see. We'd see white larvae sometimes in the stem, white or cream-colored larvae. Sometimes, and most often, we'd see a bunch of orange ones under the epidermis. And in some cases on the right, we'd see a mixture of white ones, small ones, big ones, orange ones. So we collected these larvae, put them in alcohol. Actually, I'd say I think most of this was done by Justin as far as the initial identification. And then, of course, we went at adults. So we put out corn rootworm adult emergence traps. A lot of you might have seen these. Basically, anything that comes out of the ground or plant underneath that mesh cage goes into the top of the jar through the funnel and gets trapped. As you can see, here are a couple of the adult midges that we caught. The one on the top just has his wings folded over his back, where the other one, they're held up, all right? And we caught these, but again, they have to be associated with the larvae. And at this point, heck, we didn't know if they came from the larvae, the ground, or whatever. So Justin went forward to get a direct association of larvae and adults. He had several people had several methods of doing this. This is one: you collect the adults, you get the adults isolated, and then you put them in caged pots with soybean plants growing. And lo and behold, move this path along, and you find the larvae later in that isolated pot. So I think another important thing about this was these plants were clean initially. So we did know that these adults can infest non-diseased plants initially. So just a point, we still think disease is involved somehow in some interactions, but we do know initially it can be a clean plant. And then we sent these to the specialists. Dr. Raymond Gagné in the Smithsonian Institute is a world specialist on these gall midges. He looks at morphological features, and then we sent him to Japan to Dr. Yukawa. He is a molecular geneticist, and so they looked at these, and lo and behold, both morphology and DNA did not match anything in the records. So they considered it a new species. They moved forward on this new species in Nebraska, in the Midwest here, in Iowa and South Dakota, which was kind of exciting for us entomologists. You know, usually new species are discovered elsewhere in the Amazon, but here we have them in the middle of Nebraska, well, eastern Nebraska. And they went forward and got an official ID, published that in 2019, Rosaliella maxima Gagné. And then Bob Wright went forward with the ESA, the Entomological Society of America, and got an official common name, the soybean gall midge. So now we have an insect, we've identified it, it's a new insect, so we really, except for some observations, know nothing about it. Well, let's look a little closer at this adult. Frankly, you're not going to see many of these adults with the methods we have of collecting them now. You don't collect them in sweep nets, or at least we never have. They're hard to find, but we do want you to know what it looks like. As you look at that pinny on the right, you can see from the tip of the front legs to the tip of the back legs, it's only 1/4 inch long. So these are small. But they do have banded black legs. You can kind of see this with the naked eye. I can, and I'm getting old. And then they have some mottled wings, and you can see in the middle picture, they have male and female differentiation. The female particularly, you can see that ovipositor. She uses that to lay eggs. And so you can see these with the naked eye, but a hand lens helps a lot. Now, there are other insects that look kind of like this, especially if you don't have a hand lens. So, you know, there's even a mosquito that looks kind of like this with banded legs, but it's a little bit bigger. But this is what it is with the general characteristics of the adult. Now, what do we normally see if we're in the field, like you and I, a farmer in the field? We see larvae. And we see larvae, and these are the typical and kind of general characteristics, like I said, to really see the differential characteristics for identifying it to species, a specialist is required. But basically, this is it. It's a typical maggot. It's, you know, the first and second instar, first and second stages of the larvae are white to cream colored. The third instar is what turns bright orange or bright red, some people will say. And they do look like maggots, and you know, the last instar larvae will be about 1/8 inch long there. And usually bright orange is the color. And I do want to point out again, there can be other insects that look like this. Bruce Potter and Bob Cook are going to talk about this a little bit later. You often have other types of, particularly the cream colored or white maggot in various plants that have necrotic tissue or diseases. And you can even have other insects that sequester different pigments, like this bright orange or bright red. So the identification of this insect in the field for you or me, you know, just walking in, is kind of a holistic thing. I think Bob Cook brought that out. That you kind of have to look at the symptom on the plant, where it's at, time of year, look at the insects. And if you have all these together, that lesion, these insects, you probably have got the soybean gall midge. Now, how do we work on something that's a brand new insect? Well, the first thing we do is let's look at some similar insects. So we looked in the same genus, Rosaliella, and there's like 56 species with a diverse host range. But if you look through all of them and look through the literature, one pops out as having a lot of similar characteristics and a lot of work done on it, and that's Rosaliella theobaldi, the raspberry cane midge. And although it's not a field crop type of insect like we have, it's very similar. To illustrate, you know, this insect was reported 100 years ago, and it was a significant pest since 1946. So they've done a lot of research that we can kind of look at and model after. The insects are very similar to the gall midge in certain ways. The larvae feed under the skin of the canes. The canes break easily. It's associated with fungal pathogens. And we do believe there's probably interactions with different disease pathogens with the soybean gall midge. The adults, they're short-lived. They probably have, well, they have 3+ generations. We believe that's similar probably for the soybean gall midge. The females lay eggs in different fissures and cracks of the stem, similar with the soybean gall midge. So all of these things were just, we kind of used the research that they were doing, and we kind of modified it so that we could modify it for soybean and look at the maxima, the soybean gall midge, and look at different behavioral, biological, and even management studies in order to explore this, kind of based on this other insect, the raspberry cane midge. And then, of course, we've all worked a lot on soybean and other soybean pests. So we developed some other, projects and things to work on too. Now, what were some of these projects? Well, this is an insect that comes out of the soil in the spring. So of course, we might want to look at something like planting date, which we did. Bean leaf beetle is an insect that you can somewhat manage with planting date. Again, if it's coming out of the soil, we went ahead and looked at things like tillage. We see it. Remember that first picture? We saw them along the edge of the soybean field. Well, we looked at the edge mowing, the edge of the soybean field, what's going on there. And then, of course, we wanted to look at various insecticidal, standard treatments, to try to deal with insect pests, insecticidal seed treatments, foliar insecticide treatments, at plant insecticide treatments, like, in-furrow or T-banding, things like that, and a variety of timings of these and a variety of sub-studies under each of these categories. And of course, we're also looking at adult trapping and looking at ways to further monitor this pest. And so we look at these, and actually, we're starting a lot of other experiments too. We're working with Dr. Gary Yoon, who's a pathologist, on the interaction of diseases in this insect pest. And we're also looking at things such as, the population genetics and diversity of this insect across the geographic range with Dr. Ana Velez at UNL. So we're looking at all of these things in order to find out these clues in behavior and biology, in order to transfer that to management studies, in order to try to exploit those types of things in order to manage this pest. So what's going to happen now with the rest of the seminar is that we're going to dig a little deeper in various aspects of all these studies and give you some of the results and some of the insights we've gained from the work that's been going on across this region for the last couple of years. And with that, if you do have some questions right away concerning what I've just talked about, some basic background and ID issues, go ahead and put them in the Q&A box usually at the bottom of your screen, and Justin will read those off to me. And remember, you can always just jot these down and bring these questions up later in the series, either today or in the other series in the next couple weeks. So are there any questions out there, Justin?
All right, not seeing any at the moment, Tom, but I think people are taking in a lot of, we've got a question here posted in the chat. Remember to post those in the Q&A. Are other crops at risk?
At this point, we do not believe so. We have not seen them in any extent in any other crops for sure. So I would say at this point, no, we have not discovered them in any other crops at this time.
Okay, we've got a question from Will here. Do you think this is introduced, or is it a native species that moved over to soybean?
Well, we don't know, and some of the population genetics and diversity research will help us look at that question. I tend to have a feeling that it was probably here on some legume and moved over, but we don't know that. It could have come over on another legume. Legumes have been brought into the United States for a long time, whether it's clovers or a variety of others. Some are ground covers, some are forages, some are actual crop plants. We don't know that. So that's something that we'll be looking at. My opinion is that it was here and it moved over, but that's just my opinion.
Yeah. The only thing I'd add to that, Tom, is that, we have not found this anywhere else in the world, or the two taxonomists, who know this species globally don't have any other indication that it's elsewhere. So it's possible that it's just on something that's, not well studied, and host range is a big question, but yeah, I agree with Tom. This probably came off of something else. We've got another question here. What have you seen as the highest crop loss in a field of soybean so far? So this is getting a little bit into injury, and we'll maybe tackle this one, but that'll lead to Aaron's later on when we talk about injury loss. So I guess I can briefly mention that that's a tough one. It varies year to year in terms of what we might see for losses, and Aaron and Bruce and others could comment on their experiences over the past year. And Tom, I know for East Central Nebraska, maybe I'll highlight for us, is that we can see some pretty extreme losses, although it's spatially dependent in the field. So as you move from the field edge, that is going to be the greatest potential for losses, up to 100% for a given distance. That could be 10 to up to maybe 40 to 50 feet. There may be a sporadic live plant in there, but for all intents and purposes, you may only get a bushel or two of yield. And then on out into the field, there's a gradient of more and more live plants. And so that's the most extreme circumstances. And we had well over a dozen or more situations like that, sporadic throughout the East Central part. Maybe Aaron, if you want to comment, and then Bruce, Bob, and Tom.
I'll just throw in here a minute. With some of the consultants I work with, and Justin and Bob and I work with in Nebraska, some of the early comments they were having on fields that weren't horrifically damaged like we saw some of that early picture. But when they really started to notice it, it was like, a good 15, maybe even 20 bushel loss along the first combine pass or two of the field. Then it would diminish a lot. But that is in kind of when it's typically at the edge. Like Justin said, in some really severe cases, it can go much more and quite deep into the field.
Yeah, I would add a few comments. I guess my experience is that it really depends on the field size and shape. So if you happen to have a field that's a giant rectangle, you will notice a severe yield loss at maybe one or two of the edges, and it kind of fades away as you go more into the field interior. But if you happen to have like a smaller field that's not, sort of built into the landscape a bit, you could have dead plants go throughout that, from side to side, just kind of depending on what some of the surrounding crops are, and if you happen to be near some dense vegetation. So it sort of depends on the field setup, in my experience.
Bruce and Bob, any quick comments? We got a number of other questions, but I'll address those in a second.
No, I guess in Minnesota, we don't have as many heavily infested fields as you do in Iowa or Nebraska. But we've got the worst one I've worked with is 60% on the edge, and it goes down to 10%, probably 100 yards into the field. And there wasn't many dead plants, if any, at that point, but we still had some yield loss. It's kind of an iffy estimate because we didn't have a true control. But what we see in Minnesota is it's mostly the edge effect is still pretty pronounced in most cases.
Yeah, we got a lot of good questions that have come in. All of those are going to tie into future talks. So, weather patterns. I actually copied your questions out of here. I placed them in another set, so I'm sure that we answer them when we get to those sections. Lots about rotation and management. These are excellent questions to be asking, and we appreciate them now, even because we'll be sure to think about those in a lot of detail as we move into future talks. We're going to transition to the next talk, which is Dr. Erin Hodgson. So Erin, I'll get you to share your slides and take it away.
Thank you, everyone, for joining today. I see we have over 200 people on our webinar, which is great. And I know that there's probably people from all over the Midwest, some people that have experienced soybean gall midge for a couple of years, but we also realize there's some newbies, maybe on the fringe, or some of the more eastern states who don't know what soybean midge looks like or its injury looks like. And so I think this second segment here is really going to answer some of the questions about where is soybean gall midge and some of the scouting tips. So hopefully if you're a newbie, you'll get sort of a search image for this summer, when trying to keep those positive detections coming in as new counties and maybe even new states. But also we're just hoping that with proper scouting, once you do have an established population, it's ultimately going to lead to better management. And spoiler, we don't have all the management tips yet, but the scouting is going to help inform some of that management research that we're currently conducting. So I'll be talking about distribution and scouting, and I had a lot of help with my presentation today with all the collaborators here on our webinar. And we're fairly active on Twitter, so I encourage you to keep the conversation going, especially this summer when you start maybe seeing some questionable, is it or is it not? Well, we'd like to see pictures and hear from you. So, okay, I will get started. So Tom covered a little bit of this, but I just wanted to just briefly summarize the history with that first detection in 2011. And it was scattered plants, definitely not at an economic level where people had sort of raised blood pressure thinking about this new pest, more just like, hey, what is that? And so that sort of happened in Nebraska for a few years, scattered around until 2015, where they had the first positive detection in South Dakota, followed by the first that I knew about in Iowa in 2016. These were isolated plants, definitely low levels. And it really wasn't until 2018 where we started to see more widespread infestations. We saw big patches or big strips of plant death. So we reached an economic concern. We also had the first detections in Minnesota and Missouri. So it seemed to be an issue that was spreading. And with the economic loss caused by this pest, all of us field crop entomologists started to get, really excited about maybe a new pest. And that's usually not a good thing when we get too excited. So I wanted to just share distribution. Again, this is not known to occur anywhere outside of these 5 states. And Justin put together a really nice map series showing our efforts of scouting and positive detections for soybean gall midge. So 2018 was that first year where we made more deliberate efforts for scouting, and we had a total of 63 counties in these 4 states. And you can kind of see it's kind of clustered on those state lines. And then 2019 came, and you can see basically we had 32 more counties added to the list. It's expanding in all directions, and that's largely as a result of folks like us, farmers, craft consultants, people in the ag business world who were looking, and they were spending a lot of time in beans and finding isolated plants or maybe small pockets that were starting to form. So we had a total of 95 counties in 2019. Last summer, we did a little bit more. We had a few more counties added to the list, 19, for a total of 114 counties. And you can kind of see that bubble's kind of getting bigger and bigger. And so now it includes five states and 114 counties. How quickly it might spread, how quickly that bubble will grow, we have no idea. This is a pest that is not a long-range migrator like you would think of for like soybean aphid or black cutworm. It doesn't have that long-distance migration, we don't think. And so it tends to have more local movement within fields and between fields within growing seasons. And so I would expect this bubble to grow slowly unless we have maybe some significant weather events, which we actually did have last year. But I expect this, from the top of Minnesota down to Missouri is a huge difference in how we grow soybean, the conditions, the soil, everything. But soybean gall midge doesn't really seem to have an issue with that. So I don't know what is its limitation. Is it temperature? Is it, who knows what? But I think that bubble is going to slowly expand over time. We are interested in monitoring the distribution as it spreads throughout the north central region, as we expect it to do. How fast, again, we have no idea. And so we have folks like us and other field crop entomologists in the north central region that were funded by the North Central Soybean Research Program who were looking really intensively at fields. And so we had funding to do so. And so we put a lot of sweat hours into looking for midges. And so the last summer, we had over 1,700 fields that were sampled in 267 new counties. But so far, again, it's just restricted, not known to occur anywhere outside of those 5 states at this time. So you're not finding it in other countries or other continents at this time. So what about this year and beyond? We're interested, especially if you haven't seen it before, or maybe you're not sure if you've seen it before, in new detections. We want to keep that map as accurate as possible. So hopefully with some scouting tips, you'd be able to help us out. And we'd really appreciate that. All we really do is ask for a photo because it's pretty distinctive once you see the injury. I talked about, some of those significant weather events, and maybe depending on where you're joining us from, you've heard about the derecho that happened in August. And so this is a time in the growing season when soybean gall midge adults were definitely out flying around. And because they're small, lightweight, kind of fragile flies, we think they definitely could have been picked up by some of those straight line hurricane winds that went through the majority of the states that were impacted, that already have soybean gall midge in them. So it was approximately 6 million acres that were impacted by Iowa. I'm guessing about half of that was soybean. And so we don't know if they could be picked up, how far they could move, if they could even survive those strong winds, and when they could be dumped down into the ground or dumped down back into the ground. And so we don't know if maybe we'll have a positive detection in Illinois or Indiana next year. So again, we're just really relying on people like you to help us keep that distribution map current. So no idea if it helped or hurt the midge last summer. And so I wanted to just provide some ideas about the biology so that it can help us understand first detection. So if you've never seen it before, or maybe you've only seen a couple of situations or fields with midges, I want to just provide a little bit of background so that if you really start gearing up your sampling effort this summer, you'll have some ideas. So we use those adult emergence cages that Tom showed you to try to better understand where do they overwinter and how do they overwinter. And I'd say by far, the majority of the cases, they're overwintering in soybean soil. And so they're just kind of dropping down at the end of the season, and that's where they spend the winter as a pupa. So Justin created this nice graphic, which I really like to use, so that the field on the right is, say, for example, was infested with midges in 2018. That it becomes a source. They're going to spend the winter there. And in most situations, I think most people would be planting corn the next year. So we put those traps, again, trying to find out where they overwinter, but we also wanted to know when do those overwintering adults come up and start infesting new fields of soybean. And at least in Nebraska and Iowa, that was around June 14th, 15th, 16th. So kind of right in the middle of the month, we had adults come up. So corn is up and out of the ground. Likely soybean is also growing nicely too. And so because they don't like, or it doesn't seem that corn is a suitable host, they're going to try and find soybean that current year to lay eggs and start that next generation. And so that's the currently growing soybean is a sink. And if you've ever seen the midges in person, like if you happen to see a vial or something, you're going to be really disappointed at how small and fragile they are. And we don't think they're super strong flyers on their own. And so they're only going to fly as far as they need to lay eggs and again, start that next generation. So currently growing soybean is a sink. And by far, we've had the most luck finding new detections along those field edges. So the first 4 rows that are closest to where soybean was last year, that's our best luck. And if you don't find midges in those first 4 rows, you're unlikely to find them in the field interior. They're really aggregated, that overwintering movement to those field edges. So that's where I would spend my time looking at most efficient use of your scouting time. And we haven't, I'd say we've had better luck finding the larvae on V2, V3 plants. So with 2 or 3 true trifoliates. We haven't seen larvae feeding on the cotyledon stage, or so they have to have that true leaf. And I'm not a physiologist, so I'm not exactly sure why, if it's a plant volatile or if just cracks and fissures start to form as the stem elongates and gets bigger in diameter, it becomes suitable for them to lay eggs. We're still trying to understand that, but generally it's V2, V3 and beyond is when we start seeing those midge larvae. And so again, I'm bringing up this graphic because again, if you think you have them or you're not sure, I would spend my time looking in the earliest planted fields in the neighborhood because the plants are older, they're bigger, and more likely to have midges. I would also spend my efforts looking near areas of dense vegetation. So if you happen to have a tree line, shrubs, a grassy waterway, or something like that, we tend to find them more easily than, say, wide open areas. And so again, I would concentrate my efforts on the edge, but if you had any type of vegetation near that, I would concentrate my efforts there. And then if you're not finding them in June and July, I would continue to sample throughout August, just because there would be adults moving and continuing to lay eggs during that time. So if you don't see them early, continue to scout, because you may see infestations happening later if it's a new area that hasn't been established. So in particular for the vegetative stages, if I'm walking up to a field and trying to see if there's midges there or not, I'm going to look for plants that look stressed right near the field edge. That's closest to corn. So there's going to be wilting, browning. You could even see straight up dead plants in the vegetative stage. So you're going to see little brown sticks. And so it's easy when the plants are small, because you're going to see plants that don't look very healthy. If you pull up an individual plant, you may see stems right above the soil line that are discolored, girdled. They're very weak, very bendy, flexible. And that's when you want to take a closer look, because you could confuse this field view or this plant view with fungal pathogens and other things going on. So you'd want to be able to distinguish that, but that's from the exterior, that's what you'd want to look at. Later on in the season, in the reproductive stage, again, if you're walking up to a field, you're going to notice pockets that are stressed. So they're either dead or dying, and they could be mixed in with healthy looking plants. So oftentimes it may initially look like sudden death syndrome. If you have issues with soybean cyst nematode, they look discolored, and depending on the size and the shape, you may initially think it's something else, but then it's time to take a closer look. Those stems in the reproductive stages can sometimes be swollen or cracked, but they definitely have a dark lesion, and it's oftentimes very brittle at the base, and so it becomes really weak. And so oftentimes you're able to pull those plants right from the ground, because that tissue has been destroyed and very weak. So another thing I thought was interesting this year, if you had fields where the canopy actually was closed, it is difficult to look field wide for stressed or maybe dead plants, because soybean compensates, and so it's going to fill in all those gaps. And so what you want to do is take a look and part the canopy and focus your search image to the base of the plant right above the soil line for discolorations, lesions. You may even see the larvae on the outside of the stem. And what I also learned from experience just scouting is that because the plants are very weak at the soil line, they break off. And it's kind of like the push test in corn, although not as dramatic, because you're going to hear small cracks. And that's sort of like a visual and an auditory cue that maybe that's when I need to spend a little bit more time looking. And so scouting tips just in general, Tom talked about the size and the shape of the flies, the adults, but I wouldn't even think about really trying to find those in the field. They're small, they're skittish, I think. And if you're not using the emergence cages, you're unlikely to see any of the adult flies. Even if you're using a sweep net, I don't think you'll capture them. So really you're focusing your effort on the maggots or the immature life stages. And so you would want, if you see a plant that looks like it has a dark lesion, I would either peel back the stem or use a small folding knife and split the stem and be able, hopefully, to distinguish some of the feeding injury from the maggots versus fungal pathogens, which we'll talk about a little bit later. And obviously, if you have soybean gall midge, you're going to see the maggots. I think most people with the naked eye could see those third instars because they're like little orange mandarin slices. For me, they're bright orange and they're very wiggly. And so whether they're on the inside of the stem, or maybe you can actually see them from the outside because they have basically consumed all that tissue right there. They're going to be wiggly and they're going to try and fling themselves off and away from you as you're scouting, which is always fun on a long road trip home. And so another tool that you may have access to are some of the aerial photos. So if you have a UAV with you, or if the company offers that service, this is often when people notice they have soybean gall midge for the first time, because they're out there for maybe looking at different things like fertility programs and stuff, and they see these long strips or pockets of dead and dying plants. And so an example on the left photo is where corn and soybean are butted up right next to each other. And you see along that whole interface, dead and dying plants. And so, as I mentioned with my comment earlier, sometimes you have it's a really obvious gradient of dead plants to kind of more healthy plants. And I've seen examples where, depending on your combine size, you might take 1 or 2 passes along the edge and not pick up any grain at all. It's a complete loss. Other situations, like the photo on the right, you have a long strip of dead plants that's broken up with a road and a ditch, or maybe sometimes you have grassy waterways. So they're still able to move across that. It doesn't need to be corn right next to soybean, but oftentimes wherever that interface is where you're most likely to see them for the first time. And so some final thoughts. We're interested in keeping that map current again, so please stay in touch with us this summer. If you happen to be on some of those fringe counties or fringe states, we don't know how fast they're going to spread. If you are trying to find them for the first time, focus your efforts on those corn soybean interfaces. And from June, July, and August, you have those visual cues of stress plants, and then looking for maggots inside the plant. So with that, I thank you guys for your attention. I see there's a lot of questions coming in, which is great. We're going to get to some of the management and ecology in sessions 2 and 3, but I can answer, and the group can help me with questions on distribution and scouting. So thank you.
Thanks, Aaron. So one of the questions here is kind of distribution related. The map seems to follow I-29. Does the potential movement of this equipment to new dealers and farmers aid in the movement of this pest around the area?
Ooh, I have heard that Red River Valley I-29 corridor hypothesis a couple times. I don't know if they're able to move on equipment. I have no evidence to support that. There are a lot of hitchhiking pests out there that will move with equipment, or even like soybean cyst nematode. They really talk about kind of like equipment hygiene. Does anybody else have any idea if it's supported with movement, accidental movement?
Go ahead. I was going to say, Aaron, that's kind of the north-south prevailing winds during the summertime. That's the old Puccinia pathway.
Yeah.
So it's just convenient. There's a major interstate right in the middle of it.
Next question, Aaron, is how long do the larvae last in the field? And I know we're going to be getting to this because Vilma Montenegro, a student of mine, did some research on this the past summer, but maybe a brief answer to give some hints on that.
Yeah, really Justin's crew has been doing the heavy lifting on that, but in general, I think a generation takes about a month. So you have eggs hatch, larvae feed for approximately 3 weeks, and then they pupate, and then the adult is that life stage that moves around above ground. So I'd say about a month, maybe the larvae feeding for about 3 weeks. Would you agree with me, Justin?
Yeah, and Bob Wright added a really good comment on this. He said, you'll find them from mid-June all the way through early September, and we've even found them into October, so right up to maturing soybean. I wouldn't rely on a diagnosis that late. You want to be out prior to really R7's the latest you want to be looking at soybeans, because once those plants start to dry down, that R7 stage, there's a chance they're going to exit those plants. You're not going to find them.
Yeah, it's a good point. You could be finding larvae from June, many, many months, but the actual individual larvae, we're estimating ballpark 3 weeks lifespan.
Very good questions. We appreciate you all typing in the chat and or in the question and answer and asking those. So we're going to move on. And if you got questions at the end for any one of the speakers, including Bruce and Bob here are going to speak now, you can still ask those questions. So Bruce, go ahead and take over.
All right. Bob Cook and I kind of worked on this presentation together, and actually Bob's done most of the lifting as far as publishing on one of the soybean gall midge lookalikes, the white mold gall midge. That's a picture of one in the upper right-hand corner. But I'm going to spend a little time talking about that insect and maybe some other things that might be confused with soybean gall midge out in the field. Here's a picture of the white mold gall midge, Carchimia caulicola. It's actually a relative. It's in the same family as the soybean gall midge. We've got the adult up here, and you're not going to see these in the field either, although we did catch them in emergence cages while we were trying to trap emerging soybean gall midge. Uniform gray wings, uniform gray legs, and I suppose if you're a soybean gall midge, that's quite attractive. But the adults do look quite a bit different than the soybean gall midge. These are not a plant pest. Almost all the reports of this insect, and it's been known for quite a while, have been on beans, potatoes, rape, a lot of different plants that are infested with white mold. So it's been associated with white mold for quite a while. It's through the northern continents, Europe, Russia. It was reported on a poppy in New Hampshire in 1895. Some of the poppies are actually the only cases where they didn't associate a disease with this insect. As far as we know, this is a single generation insect based on some results in Germany. It's a single generation, but the adults are around for quite a bit of time during the summer. This insect did cause some confusion, particularly in Minnesota. We started when we picked up soybean gall midge in southwest Minnesota. We were also getting reports of orange maggot in white mold infested soybeans in southeast Minnesota and up in central Minnesota. We sent some of those samples in. The damage looked different. The larvae looked a little bit different, but there was actually a misdiagnosis initially, and some of those counties were reported as soybean gall midge. Turns out they were the white mold gall midge. 2018 to 2019, we had reports from a pretty broad swath area where we had a lot of white mold in soybean. It ran from North Dakota through Minnesota into Wisconsin. And it's been, this insect or a similar insect has been reported for quite a while. Manitoba in 2014 and 16 had reports of an insect in white mold infested sunflowers and soybeans. And I've seen it in Minnesota whenever we've had a prolonged period of years where we've had white mold outbreaks. I've seen a gall midge, orange gall midge larvae associated with it. And now we finally know what it is. This insect is really tightly associated with the white mold. And, you know, from a distance, white mold and gall midge damaged soybeans maybe look a little bit similar. The distribution of white mold is spotty. It's based on where the canopy is denser. It likes it cool and wet. So high populations, areas where we've had a lot of manure, those sorts of things. The infestation happens through senescing flower petals. So you're going to see this insect, the white mold gall midge, show up a little bit later than soybean gall midge on the plants. And that distribution of the disease and the insect in the field is different than soybean gall midge. You may not see these clastic symptoms or signs of white mold, mycelia, or the sclerotia on the outside of the, or on the soybean plants. If it's prolonged, hot and dry, you can actually stop this fungal infection. And you may have some plants that were infested. You may find some white mold gall midge, even though you don't see real obvious signs of white mold anymore. So if we look at the two species, the timing's different. The Sclerotinia is not, you're not going to see the white mold gall midge until you've got the Sclerotinia or the white mold infected plants. The distribution is going to be different. It's not going to be that edge effect that you often or most often see with soybean gall midge. Those plants with white mold and the gall midge or white mold gall midge are going to be scattered through the field. And the other big key is where those maggots are on the plant. In the case of the soybean gall midge, it's going to be at the base of the plant and underneath that outer layer almost always. I'll show you an example where it's not later on. But the white mold gall midge is going to be inside the pods, inside the stem, outside the plant, wherever that white mold mycelia is. And it can be higher up on the plant. So that location of the maggots is kind of diagnostic. Without a DNA identification or a really good microscope setup, you're not going to be able to tell apart just based on larvae. The white mold gall midge tends to be a little bit paler. Sometimes there's a two-tone defect, and sometimes this head portion is a little bit pinkish, but here you can see it hanging out on the outside of a plant. Basically, if you wanted to tell these two apart and be sure that you knew that they were different, you'd have to use a microscope and look at the anterior segments of the larval abdomen. And most of you guys don't have much time to do that. The point is, you can't do this with a hand lens in the field. So if we look at the white mold gall midge, I guess it's knowledge of this insect and the distribution of it's important because first off, it's not a plant pest. This is the most commonly confused thing with white mold gall midge. Even during the summer, we've got a lot of reports from from central Minnesota of white mold and gall midge infestations. But because of the knowledge of this new other insect, the white mold gall midge, we now have a better idea of the distribution of that. We know it doesn't occur in North Dakota, at least we haven't detected it, and it's not in southeast or central Minnesota. The soybean gall midge, rather, is not in southeast or central Minnesota. And the other important reason to know that this species is out there and be able to tell the two apart is because you might want to take some management tactics against the soybean gall midge. This insect's not a pest. And it's not worth, there's no point in trying to control it. We've got some other insects that might be similar. This is actually another member of the same family. These are predators. They're going to be outside of the plant. They're going to be orange like the two species I just talked about. They're pretty efficient predators of aphids. Here's a couple larvae in a soybean aphid colony. And if you see shriveled up aphids on soybean plants, there's a good chance that this might be the insect that's causing the death and destruction out there. They feed by sucking the body fluids out of the aphids, and I don't feel bad, but it must be a horrible way to go for an aphid. Some other fly larvae, syrphid flies. These are also predators. They're going to be in the outside of the plant. They're also going to be associated with aphids. And if you've got decaying material, there's a whole host of flies that you might see. These are fungus gnat larvae. They're usually at the base of the plant. They feed on decaying organic matter. And the thing you can tell the difference here is you can't really see any segmentation like you can with the gall midges. Sorry about that. I'm afraid of my screen. Here, the other thing that we might want to look at is symptoms of the plant. And I think Aaron mentioned that this can be confused with disease. A lot of root and stem diseases can cause wilting and dying plants. This is soybean gall midge here. This is a field in 2018 we thought might have gall midge in central Minnesota. Turned out to be fusarium root rot, but there's a different pattern. We had circular areas out in the field. There wasn't any relationship to the edge, and there wasn't any relationship to previous soybean crop. Here's some of those dead plants in the background, but there's another disease that can look a lot like soybean gall midge damage other than the placement of the lesions. It's stem canker. There's actually multiple diapore species. This is stem canker. There's also pod and stem blight that can cause some stem symptoms. Those you'll often see fruiting structures in. It's rare with diapore or with stem canker. It's going to be later in the season. The infections happen at the base of the stem or at the base of the petioles. They develop these brownish red cankers. They can go inside the stem interior. And just like soybean gall midge, these this disease can cause a girdling of the stem, and then the plant above that dies, and there can be some brittle stems as a result. We had some stem canker at the base of this plant, and above this, we're getting some adventitious roots developing. Here's a typical symptom. Looks a little bit like soybean gall midge damage, but these are higher up in the plant, and there's no larvae inside. Here's another example. This is Rhizoctonia root rot. This, from pictures, we thought we had gall midge, but there was no larvae in the stem, but very similar symptoms to the soybean gall midge. Here we've got broken epidermis, and we can see a midge larvae kind of peeking through. It's easy to confuse these, but really, as Aaron mentioned, you really have to be sure you have to look for the larvae. Here's an example that we thought initially was pod and stem blight, stem canker when we were in the field. All these nodes here had a discoloration, but when we peeled these back, there was maggots, orange maggots inside. This was there's a couple plants mixed in with other soybean gall midge infested plants. So for some reason, the adults were finding a spot to lay eggs here. And these actually got into the interior of the plant, which is very, very seldom, I've seen very seldom with the gall midge, soybean gall midge. All right, so overall, there's a few insects that could be confused. The biggest, easiest to confuse is going to be that white mold gall midge. But and there's also some mechanical injuries, some diseases that can cause stem symptoms similar to the soybean gall midge. But if you look at where the symptoms are, what the symptoms look like, how that damage presents itself in the field, and how the larvae, where the larvae are at the plant, those are going to provide some diagnostic clues. So that's what I've got. Here's some more pictures of soybean white mold gall midge, and just kind of the variants of where it appears on the plant. Here you can see a plant where most of the mycelium is already gone, but there's still some maggots hanging out on that stem. So, thank you.
Yeah, thanks, Bruce, and great overview to all the complications that we face. I know each year I get a number of adult species submitted to me of various things. So I think there's people are eager to find adults, and we're just not likely to see those. So really good overview, especially on the disease side, which is an ongoing question with soybean gall midge. We have a question that could be to any one of the group that I'm pretty certain we don't have any really good answer to, but it's a very good question. In the future sessions, will you cover density dependent factors that you've been able to detect affecting the distribution of and this pest being a marginal pest? What might be some of those factors? That is an excellent question, loaded full of a lot of years of research, I think. But does anybody have any initial thoughts they want to add to that question? I think we all have a similar kind of response because it's loaded with a lot of ecology, biology related information, and certainly is something that I think we'll address in the coming years as we learn more about soybean gall midge. We are eagerly pursuing, and as part of NCSRP funding, looking at the distribution of this insect. And one of our goals with that is where we see extreme pressure is to look at the landscape, crop characteristics, and other things that might be driving this insect to be more of a problem in some areas. So that's the abiotic factors we may be able to isolate in the future if things appear clear. Those other things, those density dependent things are on our radar, certainly. Bob Cook is here. We're looking at biological control and trying to look at the funding side of that is tricky, but there's opportunity to pursue that side, which is an important part of the equation given the difficulty we're facing with chemical control, as we'll be discussing in later sessions. So 4 lines of text, a long, lengthy answer, I think that we don't have a lot of information for right now. Anybody want to add anything to that?
Well, I'll just say, looking at the distribution, we need some answers of distribution in the field and things like that also to answer those questions. And that has been difficult too. Like with the, we monitor, all of us have traps out in the field early in the spring. And what is interesting is sometimes we'll have fields that later on, we'll see lots of midges next to it emerging in the field next to it. Yet when we have the emergence cages out, sometimes we're catching a lot, sometimes we're not. They seem, they're aggregated often, in spots in the field, but it's difficult sometimes to get those answers of distribution in the field to answer some of those density dependent questions. All I know is early when we first get fields that start out early, it starts out on the edge. And when you get bad years, it'll have problems further deeper into the field. But then that seems to be a function of generations moving on, and then moving deeper in the field too. And then like Aaron was saying, the, features and landscape features of that field, waterways and such. But that is a difficult, getting that base, we're having, it's difficult to get some of that basic distribution data to where we want it.
Yeah, I would add a couple of things. The fields that I set up, and we talked about, farmers being very gracious to let us set up research. Fields that I had sampled in 2019 that were hit really hard. I was surprised to learn that revisiting their farms in 2020, the infestations were a lot lighter. And so in addition to some of the density dependent questions that were asked earlier, also the seasonal dynamics, even within a relatively small geographic area, are highly variable. So in addition to not understanding density dependent populations, also between seasons. And so we see that with a lot of pests where you have really high years, you have low years. And so there's so many questions about the dynamics that we don't understand. And I don't know if any others have things to add about that.
Just on the plant to plant, how many per plant at what plant stage cause the plant to have issues, whether it's death to, physiological yield loss. We're exploring that too. Just how many insects per plant cause a problem and at what stage, because you can have these infest the plants at various stages throughout the season.
Any other comments on that? I know
other than it's actually possible that some of these heavily infested fields, the midges do so much damage that they basically starve themselves out. There's a lot of questions that we just don't have answers to yet.
Yeah, I'll provide a bit of hope. We just got USDA funding to address the distribution spatially of overwintering cocoons. And so part of this is to look at this injury level scale in the field and say exactly to Bruce's point, which is, are they taking out their own environment to where they limit their ability to overwinter in a given area? And so we'll learn a lot more about that. Randy Warner, who I know quite well, is a farmer that I've worked with, just posted a question. Him and I have had a number of conversations about this. And he asked about sweet clover because we are well aware that sweet clover is a host. And that's a complicated thing because Tom started off talking about orange larvae, their presence. Bruce covered the potential other things that we can find that are similar. And it's quite possible that on sweet clover, there may be other species that are present that we are unaware of because this genus has not received a lot of attention. Although we've done, much like Tom discussed, the Koch's postulates of drawing that through and onto soybeans, and have successfully done that to know that it is the same species in genetics. There is one other one, alfalfa, and the sweet clover has been documented in Iowa with Aaron and in Minnesota with Bruce and others. And so we know it's present in those areas, and there's a lot of genetic work to be done. The other one of that is alfalfa, and that may raise alarm bells with other species. Don't raise alarm bells on either one yet. We don't know their ecological importance in terms of causing a new infestation. We'll cover that in the ecology section. But these are things, I think, in new areas where you might want to look at those. We'd be happy to get a sample that would help inform us and help us understand this insect's differences genetically on those hosts in geographic regions. So good question, Randy. We've got another one. Is anyone looking at host plant volatile attraction? How are the midges finding these plants? These are a lot of ecology related questions. We appreciate those. I will copy that question and paste it into my list of questions that we'll start to address. I can tell you, we won't have a distinct answer for soybean gall midge. We just don't know a lot of that yet. I mean, all of us know, looking under a scope at the adults, they have very large eyes. And it's apparent that visual cues may be part of what they're using, but the specific cues, some other species lend some opportunity. But this is, we talked about the others briefly. Unfortunately, not a lot of those are all perennial systems. We're looking at an annual rotated system where movement is a lot different. And so it lends cues, but it also causes some problems for us. So good question. We'll try and address it on the 12th. Other comments or questions that anybody wants to discuss? We're 5 minutes over, so I appreciate those of you hanging out and asking questions. And of course, if any of you want to try and address that question, go for it. All right. So if you're muted and trying to talk or writing, hold on to that question for the next session. We appreciate your time today. Be sure to join us in the next two. This is, of course, recorded. A couple of you asked about that. We will do the closed captioning, so that will take some time. It'll be available on the University of Minnesota's website, as well as we'll put this up on the soybeangallmidge.org website. Enjoy the rest of your week. Thank you for joining.